there's going to be a transformation, a normalization process. So if you provide something normalized, re-normalize something, it doesn't make any sense. Our metadata that can be, for now, we only work with one column, but we can have multiple columns, and that should be the topic. I don't know if we're going to work with this tomorrow or not, or we close the topic now, but we can have multiple columns and work with multiple factors. But we just work with one today, right? We start with a condition. We was comparing normal with the sleep deprived. Then we use a different column, but still it was only one column that had 4 factors. So when we give the design, what we're asking is column names of our metadata, right? So we want to study, is the condition the main source? So we're saying the expression differences we observe are based on the condition. So, and the sig2, oh, I have this. Right, we provide the 3 pieces of information, and what we were testing is that, right? So if this difference was significant or not, different from 0 or not. And this was I actually picked the gene with the highest difference in this case, because it was pretty obvious. So we think about it, what we provided this design. We've seen the output table. I guess I was a little bit fast, but I'm going to repeat again. So this is just the average across all samples, the log2 change, the standard error of the photo p-value and p-adjusted value. Questions about this? The problem came when we had multiple factors within a condition, right? And then in those cases, what we did is to provide the contrast to access the other pairwise comparisons. So the function of contrast is to access the other pairwise comparisons. If you don't use contrast, what DSig2 is going to do is to find the first comparison and give you just one. When you ask for the results, it just gives you one pairwise comparison, not all of them. So the way to access it is to tell me, we tell it, please, from the column that I'm working with, compare this with this. That's what we said, right? So that's the way to access other comparisons that are not present by default. Because DSig2 only returns one table at a time. So if we have multiple treatments, imagine we have treatment 1, 2, and 3, and 4, you just keep it in a single column too, and you need to do pairwise comparisons. That could be an option to do the analysis, right? So I show you the model matrix. I explain quickly, again, for this type of comparisons, even if more complex, you don't need to access the model matrix, but you could, right? So this is how it's aligned. And basically, it's telling you the grouping that we're going to be using and how internally it calculates the coefficients. So to calculate the baseline, it's only going to use the normal samples, right? So it doesn't use the condition slip. So it calculates the baseline. Sorry. Oops. Calculates the baseline. If we could plot an arrow from here to the baseline, and that would be the coefficient or the baseline or the intercept. And the other one has the baseline plus something else, and we are estimating this. That's how internally it's making the calculations. And I'm trying to plot this because if we do, that's what I was saying, if we do more complex models, it's good to visualize to understand what coefficients this is calculating. Most of the time, you don't need to do this because it's easy to understand just looking at the formula. But I don't know what you're going to be working with. If you start working with 3 or 4 factors, each one of them has 4 or 5 levels, it gets really complicated. What factor or what coefficients DSig2 is using or calculating. So we give you this example, right, yesterday. And thanks to the PCA, we observed that, you remember that example I gave you the PCA with the mice in cages? There was this train, there were...
So if comparing multiple factors, how is the coefficient, will the coefficient be different for each comparison, or it will take a mean of everything?
No, so what DSig2, let's imagine we give them this formula, right? So we're using different factors here. We're using the strain, the gender, and the condition. for simplicity, let's assume there's only 2 of them in each one of them, right? So we have only 2 genotypes. We only have 2 genders, I guess. And we can have 2 conditions. So DSeq2 is calculating only 3 coefficients. No, it's not doing anything else. It's calculating 1 for condition, 1 for gender, and 1 for strain. And we can talk about this next, I think. OK. So somebody was asking about batch effects. So when you work with batch effects, then is when you start getting more complex formulas, right? Let's imagine this situation. We have a condition, normal versus a KO. You have this situation, but you realize that within each of these conditions were analyzed by 2 different labs. And you suspect there could be a batch effect. Actually, when you plot the same thing and you separate by lab, you notice that there's some sort of batch effect here. You see that the condition changes are similar, but they're at different levels. So this is a classical batch effect situation, right? Your baseline, your normal, is different from lab A than from lab B. If we analyze this, there's a lot of noise here. We may be missing genes, right, that could be differentially expressed, significantly differentially expressed because of the noise generated by the lab. That's a batch effect. So there are ways to deal with it. You can use tools to remove batch effects, right, which would modify your data. So you can use Lima, which is another R packages that can deal with batch effects. Or you can model it, right? You can add it to your model. What I recommend, I don't know. I prefer the model idea. For whatever reason, I don't know. I mean, the word batch, you can define it in different ways. We think batch effects something technical, right? Which is the case here. The lab is not something that we care. It's not part of our question. We kind of want to remove it. There's a preference about modeling or removing. I don't know. I like the idea of modeling better than removing, but I don't see a problem either, right? You can use Lima to remove the batch effect and keep doing the analysis. If you want to be working with DSeq2, it seems easier to just model it. It allows for that, right? So actually we have, so this blue line, as you can see, is the actual effect we want to study, but there's some effect also caused by the lab. Yep. Yeah, and this is what we generally call batch effects, right? But I mean, I don't know. There's also biological effects that can batches, right? We have the batch of being normal and the batch of being KO. But generally, when we think of batch effects, it's about technical issues. So now we have a new metadata, right? So we have 2 columns. So we could formulate it like this, right? So our expression depends on the batch and the condition, because we notice that it has an effect. So if we do the model matrix, now, again, we have an extra column. We can see the classification between them. So now it changes a bit. We have a new baseline, right? So the new baseline is the normal one based on how we build it. We can build it differently. The baseline could be different, but the way we have it here in the table, the one that has zeros everywhere except for intercept is normal lab 1. So our baseline is this. Yep. So now, this is what DSeq2 is going to do. The first coefficient is going to calculate is the next one, which is the one for batch lab B, based on this thing, right? So the one that only has a 1 here and 0 everywhere else are these samples here. So it's calculating, OK, this is a coefficient I'm going to calculate. So from the baseline to the batch effect of being normally lab B. Now, the next coefficient I'm going to calculate is this one here. The only one that has zeros everywhere and only ones here is this one. So this is the effect of the condition. So that is more or less how internally is going to work. I know you don't care about these things, because basically what we're saying here is give me two coefficients, one for the condition and one for the batch. I only care about the condition. So I'm only going to ask you, give me that coefficient. And by default, if you build the design formula like this, dsig2 only returns condition. If you ask for the result table, it's only going to return your condition, not the batch. So what you're doing effectively is removing the batch effect to some extent. If you don't ask for it, if you don't use contrast, you're not going to get it. So the way to get the red line, I don't know if you care, but if you care, you need to do contrast, batch, compare this versus this, and it will return you the red coefficient here. But if not, it's only going to return blue. That's by default how dsig2 works. So if you don't want to think about how these things are built, basically the question you're asking with this formula is, what are the differences between KO and normal regardless batch? That's what you're asking. Yep. So if I switch batch by condition, if I say condition plus batch, dsig2 by default will return the red one and not the blue. So when you build the formula, if you want to deal with multiple things, put at the end what you care. So if we have, this can be as complex, I think I'm giving an example more complex than that. Well, like, here's what I'm saying. By default, a dsig2 is going to print only this. So I'm giving an example here. So if we ask for the coefficients, they are all calculated, right? If you ask for that table we saw before, the coefficients table, you're going to obtain all the coefficients. But if you ask for the results, you only get the ones, the condition ones. So if you really want to access what is the effect of the batch, you need to use the contrast to ask. If not, you don't get it. Yep. So, oh, you can, it's not going to show up, right? Because we have the problem. But I mean, I guess I can ask this. If we want, it's showing up, yeah, but it doesn't show live here because it's asking, the presentation is not live for whatever reason. I need to do that trick. I mean, we can do it. I mean, we can try. Let's see. Oh, no, my bad. I actually need to start it. So can you build a design formula to answer this? So build a design formula in a way that my question that I'm trying to answer is, regardless of the genotype, I don't care if it's normal or Shank3, I want to know the effect of being sleep deprived or the effect of condition. So regardless of genotype, I want the condition. Based on what just tell you, what is the design? Can you, don't be afraid of typing, but don't use white spaces every in a single word.
Baseline is the normal here.
Don't worry about the baseline. You don't need to know what's the baseline. But yes, I mean, if I do the level thing, I'm going to say that this is the baseline. And this is the baseline for genotype, and this is the baseline for condition. So each column can have their own baseline. Or, well, if you think about with the graphic that I show you, it depends on how you build the formula again. So my question is, what is the effect of condition regardless of the genotype? Remove the genotype effect and just give me the effect of the being sleep deprived. How is the formula you're going to be using? Is people typing? Are you guys typing things? Even if it's wrong, just type something and see if it's live.
So it's not updating.
It's not updating? Nothing. Well, if it's not updating, we can keep going. I'll give you the answer. So if your question was that, right? So I only care about the difference in the condition. I don't care about the genotype. And I want to remove the effect of the genotype. As I told you, put the last element of your formula, the one that you care. So I care about the condition. I don't care about the genotype. And by default, dseek2 will return the effect of the condition and not the genotype. If I only use one column, right, I get mixed up these effects. So I'm not calculating the genotype effect. If I use both, I'm splitting the variance on the data, right, into two factors.
Why we need to add genotype? We will give only design is equal to metadata and condition.
Okay, so if you only use the condition column, you are mixing the genotype effect in your formula. So if I remove this, right, and I'm only using a column, the genotype effect is going to be mixed up with the condition because I'm not giving this information. If I want to separate both and calculate both, I can use this formulation and keep them separated, but I'm only reporting the one that I care. Yes? I don't know. Your faces doesn't look like you. I'm probably not doing a good job. I don't know if there's a different way to explain it. Verbally is the best way that I found to explain these things. When you build your formula.
Kept this constant. Genotype is now constant.
Genotype is constant. No.
But it's not accounting, right?
So it's this problem we had before, right? If I go backwards.
What I mainly want to see, that is coming at the end. I want to see the effect of treatment that I put at the end.
Yes.
dseek will take that as a primary motive to analyze the data. But
So
at the same time, I don't want to miss out the effect of the genotype.
because if you don't, you have this. This situation is the same one as the genotype, but if you call genotype A, genotype B, you are in the same situation, right? So you prefer this situation where you clearly can separate the effect of genotype and condition than working with only one column without not knowing what the genotype is doing, right? So that's what I'm trying to explain. So the batch effect situation is identical that the other one that we talk here, right? It's exactly the same thing. The only difference is I call it with a different name, but this call it batch.
So, if you actually take a moment away and then from mathematics of it, think about what is the biological question you are trying to address here. What is the biological question you are addressing?
Treated versus non-treated.
No, just tell me in one sentence what is the biological question we are. We are addressing here that if SHANK3 knockouts behave differently in wild type, sleep deprived and normal, if there is any difference or effect of SHANK3 mutation in sleep deprivation.
But hold on. No.
That is not the motive.
So no, hold on. Your question comes later on. The question I'm trying to answer here is, what is the effect of the condition regardless of the genotype?
No, that is fine. But the issue here is that in the data, there are two components of the data. One is the genotype, which is loss and gain of SHANK3. And we understand that anything what you do with the rest of it, sleep deprivation, otherwise, there might be some effect. That is the null hypothesis. So genotype needs to be taken into account in your model, unless you will not get that, because that is the null hypothesis. And if you want to really to see that null hypothesis or accept or reject the gene without the genotype in your equation, you will not be able to see that.
Yep.
I have that group
So
in my
what really will happen?
Yeah, use it.
I want to use it.
Yeah.
No. So if you are actually just using condition, then what will happen? The genotype will confound your.
Yeah.
No, I want to see that also. I
Yeah.
have put the condition in my experimental design. I don't want to miss out on my genotype as well.
Oh, sure. I mean, you don't miss it if you do this, because you use contrast and you get it. What I'm trying to say is, I'm just hypothesizing if your question is the one on top, how you would formulate it. I know, and you can change this word by batch, you can change whatever. Yep. I think you got it now, right? I think we got in a better position. So I think it's good if you formulate your question in paper to understand how you do the formula. And
I think we should stop for so that we can set up.
Yeah, so because it's going to, the next question is a bit different, but I think we can leave it here.
We'll continue tomorrow also, I
Yep.
think. And one, there is a suggestion, and it all depends upon all of you. So the suggestion here is, we have, the multivariate analysis also, the multi-factor analysis was also there, which we can discuss some more if you want. So if you want to spend some more time tomorrow, we can start early and end late.
We can do half an hour tomorrow. We finish this and then we move on.
Saying like if you want to start early, like really 8 o'clock, but everybody needs
8:30, 8:30.
And yeah, but then 8:30 means 8:30. Everybody should be there at 8.
No, we'll start at 8.
So,
And we start at 8:30 because today it was 9 when we started.
then actually you want to stay a little later also, then we can continue, because if you want to go through Friday is the only day we are left with variant analysis, so that means our RNSC, single cell bulk, all that discussion needs to be completed by tomorrow. So it's all about, like, if you guys are ready to work a little harder tomorrow, then one day, a little more effort, then fine. Otherwise, it's okay.
I know. So at least we're going to do an exercise about formulating questions and how you put it in a formula. So I'm going to try to forget a little bit. I know this sounds, drawing lines like this, it sounds weird now. I'm happy to explain it better, but I think phrasing helps a lot too. And knowing what DC2 does with that formula.
Translate. Can you introduce Rick?
No.
Please do that.
No.
He is your friend.
Nope.
Give him some churros, he will then introduce.
No. We
Can you hear us? Sorry.
You're muted, Tony.
I'm muted? Really? Can you hear me?
I can hear you. Can you hear me?
Yes, I can hear you.
Excellent.
Can you hear me?
I can, yes.
Okay, great. Okay, so let's
Hey, my name is Rick. I'm in the Department of Pediatrics, section of critical care, and I run a research laboratory that's focused on blood vessel dysfunction in critically ill children. And the goal of my research laboratory is to understand and treat how blood vessels stop working in critically ill children. And we're really focused on how endothelial cell junctions come together. And we'll get into that a little bit more later. But the outline of the talk is this. I will discuss 3 projects in my laboratory that involve next generation sequencing. And they all involve samples taken directly from critically ill children. And if we have questions or want to talk about the particulars of the next generation sequencing strategies, I'll defer to Boney, because all of these projects, I've worked with Boney and Fransek to do these. So the first thing I'll talk about is how we get patient samples and use next generation sequencing to identify drivers of pulmonary capillary dysfunction in critically ill children with viral-induced lung injury, termed bronchiolitis. And I'll talk about how our sequencing results directed some of our experiments that we did and show you some of the findings we have with that. And then we'll talk about how we use next generation sequencing and single cell RNA sequencing to identify mechanisms of blood vessel breakdown in critically ill children with sepsis and shock. And so that's a different set of patient samples, a different set of sequencing, and a different set of results. And then finally, if there's time, I talk about how we've been working with Boney most recently to isolate the DNA in the blood of critically ill children to see if we can do sequencing of methylation profiles in that blood and use that to identify biomarkers for the duration or severity of multiple organ dysfunction syndrome. And I'll take questions at the end of each one of these little mini projects. So just by a way of background a little bit, I study endothelial cells, which are these cells here in brown, and they are the cells that form the inner lining of all of the blood vessels and do really, really important things. They help regulate blood flow and oxygen delivery to tissues. They help regulate clotting. But what I'm most interested in is and life depends on keeping that blood and that blood volume in your blood vessels and not leaking out. And if ever you've had a family member or friend become critically ill, you may notice that they become swollen, and that's because some of that volume, that plasma volume, is leaking from inside the blood vessel space where it's helpful and provides oxygen delivery to outside the blood vessel space where it causes swelling. And if their face and hands and feet are swollen, then their organs may be swollen, and swollen organs aren't going to work as well as non-swollen organs. So this is what I'm working on here is the intersection of permeability and inflammation in critically ill children. That's a major focus of my lab. So project 1 is to identify the drivers of pulmonary edema in children with viral lung injury. We call viral lung injury bronchiolitis. And in America, this is a major problem. Viral lung injury is the #1 reason that kids come to see their doctor unexpectedly. It's the #1 reason kids go to the hospital, and it's the #1 reason kids end up in intensive care. And we don't have any medicines to treat the virus or the viral lung injury. It's only supportive care, including help breathing. And we can put on a nasal cannula or a mask on their mouth or put in a breathing tube if the lung injury is severe. And there can be other injuries too, like we talk about viral injury, but there can be bacterial infection, trauma, ischemia reperfusion, or systemic inflammation that can also cause lung injury and trouble breathing. And fortunately, most of the disease is mild, but some is severe. And when severe disease, we get x-rays like this. So this is a chest x-ray of a child who has severe lung injury. And you see this x-ray, you see these ribs, this big thing in the middle here is the heart, and then these are the muscles of breathing, the diaphragm, and this is some air in the intestine here, and this big thing here is the liver. But this should be air. Your lungs should be filled with air, and you see it's not. It's filled with all this white, hazy cloud stuff. That's fluid. That's fluid that's leaked from inside the blood vessel space to outside the blood vessel space. And fluid that leaks into the lung causes three main problems. First is there's a very thin air-blood interface. It's only 3 proteins thick, actually. And when that barrier gets swollen, that's more distance for oxygen to diffuse. So kids have trouble getting oxygen into their bloodstream. And then it takes more work to breathe with wet lungs. Like, think about moving a wet towel versus a dry towel. It's a lot harder to breathe in and out if the lungs are filled with fluid. And then, you know, if their alveoli or lung spaces have fluid in them and they collapse, then the blood that's going to those won't participate won't get oxygen or drop off carbon dioxide. And so, you know, we call that air blood flow matching. And normally in kids that's pretty well matched, but if fluid filled with alveoli and collapse, then they have worse air and blood flow matching. And so all of these processes lead to like really bad hypoxemia or low blood oxygen level from the pulmonary edema from blood vessel dysfunction. And so we wanted to sample the processes that were happening here, but it's very difficult to get this. So on this child has a breathing tube right here. Maybe we can use that breathing tube to sample some of the cells or the processes that's going on here. And they're very difficult, right? Because there's so many cells throughout the airways. This is kind of the small airways coming down. There's epithelial cells that are secreting molecules. And then there's alveolar cells. And this is where the air exchange happens with the blood, is in the alveolus. And so there's all these different types of cells that are secreting all these different molecules. And we want to study all of it. And so we made a protocol to look at both the lung fluid that we'll send for metabolomics, proteomics, and functional assays, and the lung cells, which will do single-cell RNA sequencing and next-generation sequencing. And so this is the goal, is to survey all of it. And how do we do that? Well, we have a protocol that starts at the bedside. And this is a critically ill child intubated for lung injury. And we pass a suction catheter down this ET tube here, put some saline in, and suck it all out, and get a sample that we immediately process for multiple different things. And so the first thing we look at is single-cell RNA sequencing. I'll talk about that. We also send these samples for proteomics and look at a wide range of proteins. We look at metabolomic profiles of these cells, and then I'll explain a little bit about the functional assays we use, and these are cultured endothelial cells, and we can test the effects of the patient samples on barrier function in cultured endothelial cells. So I'll start with the single-cell RNA sequencing. We use 10x genomics to look at the children with lung injuries cells, and we get a very, very good breakdown of different cell types. We see a large population of monocytes. We see some alveolar epithelial cells, which is great, some bronchial epithelial cells, macrophages, some T cells, and some other cells in the lung. And I'll show you a little bit of how we analyze what these cells are doing over time in kids with lung infection. But something really neat that we can do is we can integrate the viral genomes into the reference genome when we process the single cell injury, and we can see, this is for RSV virus, respiratory syncytia virus, we can see what cells are infected. And it's mostly the alveolar epithelium and the monocytes, and not so much the other cells, which is what we knew, but it's good that we confirm that. And so we actually have expanded this project to look across 10 centers in the United States to get these samples, and we're going to look at the prevalence of different viruses in different cells using this really cool sequencing technology and analytic technology. So I told you we also do other things on these cells. We send them for metabolomics, and metabolomics, really show different profiles. And so in pink here are samples from the children with lung injury, and in the green here are samples without lung injury. And when you cluster the metabolomics, they essentially have orthogonal profiles. And this is telling us that there are very different processes affecting the composition of the fluid in the lungs from healthy children compared to controls. Now, we can also get samples over time. That is, look at the same child when they're sick with lung injury and when they're getting better. And when we do that, we see that there are, the children are starting to recover. So these are children that have recovered from lung injury right before the breathing tube is pulled out, and it seems to much better match the profile of the healthy control children. So these metabolomics are telling us that there may be some changes in the lung fluid that are relevant, and we wanted to look and see if we could find out what proteins may be driving those changes. And so we sent the fluid for proteomics, and we see lots of changes. So this is comparing children with bronchiolitis compared to healthy children. We see a lot of proteins that are elevated in bronchiolitis and a lot that are elevated in healthy children. When we look at bronchiolitis patients that are sick compared to when they get better, we see somewhat less proteins because it's the same child, so it's a little bit better controlled. But we see some similarities, and the similarities we see are IL-1 signaling molecules. Endogenous IL-1 inhibitors like IL-1R2 and IL-1RA. These suggest that IL-1 signaling is dysregulated in children with lung injury. And so we went back to the RNA sequencing to look for that and found that in fact IL-1, the molecule, IL-1 alpha, IL-1 beta, as well as all the IL-1 endogenous inhibitors are much higher in the virus group, that's this purple bar here, compared to the cells isolated from the healthy control children shown in green. So this suggested that IL-1 and IL-1 inhibitory proteins may be playing a significant role in the pathogenesis of lung injury. Now, IL-1 is very well studied in endothelial cells, and it's very well known to cause barrier dysfunction or leak in endothelial cells. And so we wanted to study that using cultured endothelial cells. And what we use is this setup here, where we have a 96-well plate, and we grow endothelial cells on culture with these gold electrodes. And we can measure the electrical current flow between those electrodes. And so we get things that look like this, where the cell layer that we grow on this becomes a resistor, and then we apply Ohm's law and we can measure the current flow between those cells. And that gives us a measure of barrier function, how well this endothelial layer is resisting electrical current flow. We can measure that, and we measure that in graphs that look like this, where we have on the y-axis a relative tier or relative transendothelial electrical resistance. That's how well the endothelial layer is resisting current flow. You can think of it as relative barrier function. And then on the x-axis we have time, and we get graphs that look like this, where if the bar goes up, that's better barriers or less leak, and if the graph goes down, that's worse barriers or more leak. And when we look at patient samples, we see that there are in fact profound effects. So on the left here is from one patient with RSV, and the solid line is from when the patient was extubated or had the breathing tube removed, meaning their lung injury was better, and the dotted line is from when their lungs were sick or when the breathing tube was placed. And you can see that the dotted line goes down way below the solid line, suggesting that the lung fluid from when the child was sick induces much more barrier dysfunction than when the child was well. And the same is true for this is the second patient in red shown here. We see the same effect, that the lung fluid from children when their lungs are injured induces cultured pulmonary capillary barrier dysfunction. And so we wanted to look at this, and we can do confocal microscopy on these cells, and we see things that look like this. These are cells that we've grown and treated with saline. And so here on the top in the white is F-actin. You see it forms nice, smooth ring around the cells. And then here is a junctional molecule, a tight junction molecule called ZO1, zona occludens 1. And it also forms nice, smooth barriers. And then on the bottom here is the merge, where they're together. You see there's nice, smooth outline of endothelial cells. Hopefully you agree it looks quite pretty. When we put patient samples on them, we use 10% of the patient lung fluid. You see there start to be actin stress fibers forming. There's punctate breakdown of these junctional molecules, so there's interruptions of that smooth staining, and the molecule and the merge looks less happy. These cells are forming fibers to start to pull apart. We know that IL-1 causes cells to pull apart, and we're starting to see that here with stress fiber formation and junctional problems. And when we add inhibitors, we add the endogenous inhibitors to the patient sera, we see that it's better. So here is the same 10% patient volume, but we added 100 nanograms per ml of IL-1RN, and here we did IL-1R2. These are both soluble molecules that bind IL-1 and don't allow it to signal in the endothelial cells, and you see that there is preserved staining, smooth staining. There's less stress fibers, and the merged cells look better, look like they're pulling apart less. And we can quantify this by looking at the smooth junctional staining. And when we do that, we see that really the cells treated with IL-1 RN, shown here, have significantly less barrier dysfunction than those treated with the 10% sample. And you can see that visually here at the bottom, where these have less well junctions and more stress fibers, and these cells look a little bit more confluent and happy. So that's where we're at with Project 1. Hopefully I've shown you that next generation sequencing can identify targets that we can study in culture, and that these targets are immediately relevant. They're relevant because the kids have pulmonary capillary leak, and we're doing sequencing on those samples. And that sequencing is supported by a multi-omic analysis with protein and metabolomics that support these changes, and we can test that with cultured endothelial cells. So with that, I will pause quickly and see if there are any questions.
Rick, can you hear
You're
me?
muted if you're trying to talk to me.
So can you
There it
hear me?
goes.
Can you hear me, Rick?
I can, yes.
So I have a couple of questions. So do you get enough endothelial cells in your BAL samples to do the downstream experiments? And the other one is regarding, and the other one is...
Yeah, so we unfortunately don't get any endothelial cells. We don't get any endothelial cells. These are all cells that are in the lung fluid, and we think that these cells are secreting these molecules. These molecules are going to diffuse back to the endothelial cells, which would be here.
But for the cultured endothelial cells, where do you get the sample from?
Yeah, so we get those. That's a great question. Those are these are primary human microvascular pulmonary cells.
Okay. These are HUVEC cells, and you're treating them with patients' BAL samples, right?
Not HUVEC cells, not HUVEC.
Okay.
HUVECs are isolated from umbilical veins, from umbilical cords. We isolate these cells on our own from healthy lung tissue. So we get healthy lung tissue that's
Okay.
like resected for whatever reason from the Yale pathology people,
Okay.
and we isolate capillary endothelial cells from the lung. You can also buy them. They're called HLMEC, or human lung microvascular endothelial cells from Lonza, or we call them HPMEC, human pulmonary microvascular endothelial cells.
And the
But they're
other,
primary cells, they're
Okay.
human, and they're from the lung.
okay. And the other one is like when you say that IL-1 is increased, so have you tried IL-1 blockers in any of these patients? Or, I mean, empirically, have IL-1 blockers been useful in patients who have got this severe bronchiolitis? Has it been reported of any use? Because your data suggests that, and your experimental data also suggests that blocking IL-1 by this antagonist or would be of some use here.
Yeah, so that's a phenomenal question. You and I are thinking exactly the same way. I mentioned this big grant that we just got to look across 8 centers. We're going to test the hypothesis that IL-1 is driving pulmonary capillary leak. So we'll have more data. You know, this was just all from like 3 or 4 patients. So we'll have more data to support that. And aim 3 of that grant is to test the IL-1 inhibitors in a mouse model of bronchiolitis.
Okay.
And what's great is that IL-1RN is FDA approved for use in kids. The name of it is anakinra.
Yeah.
And so it's already being used in kids with like severe rheumatoid arthritis or other really inflammatory conditions. And what the point of our grant will do, hopefully, will support what exactly you said, that we could actually deliver IL-1RN to the lungs of critically ill children, and that would be the first therapy specifically for pulmonary capillary leak in lung injury.
And
And it's even better. It's even better because in kids we can nebulize, like we can nebulize albuterol, and kids breathe it in right to their lungs. We can nebulize this protein, so we're delivering it only to the lungs, which means it will have a better, more precise effect and less off-target effects. So the answer hopefully is yes. We're working on it.
okay. So the other one is related to like, is the response different? Are the data different on patients, the children who have been immunized? Do you have like vaccinized? Like,
Yeah, we in our small patients, the data I showed you, we don't have their vaccine records.
Okay.
In the larger study, we'll be able to have their vaccine records, and we'll be able to know exactly what virus they have, because we'll incorporate many viruses. Like we'll incorporate, the one I showed you was just RSV. We'll put in all of the viruses that affect kids into the reference genome. And so we should be able to look at that, but we may need even more patients than we're trying to enroll, because that would be a harder question to ask.
okay. Yeah, that's all. Thank you. Nice work.
Great, thank you. All right, really great questions. All right, so we'll move on now to Project 2. And Project 2 has been like a super big effort. Like I've been working on this project for many years, and it's one of those things that if we knew how hard it was when we started, we probably wouldn't have done it. But we have, and we have great results. And so the point of this is to identify mechanisms of blood vessel breakdown in kids with sepsis. And to do that, we needed to look at the endothelial cells. We needed to find a way to see how the endothelial cells in critically ill kids are different or changing from generally healthy kids. And this was really hard to figure out. How are we going to get endothelial cells from critically ill children? But we found a way. And the way is with the equipment we use to put in central lines in kids that are critically ill. So when a child comes into my pediatric intensive care unit and they're very critically ill, they get a large IV called a central line, and it goes in the veins of the neck or the groin. And we put it in with like guide wires and equipment, and so we're able to look at the endothelial cells that get stuck to the guide wires that we use to place the central lines. Because the guide wires are inserted through something called the Seldinger technique, and they bump up against the vein. And so when they take it out, we grab the wire and we run off and do single cell RNA sequencing. And we compare critically ill children in the PICU, this second bullet point, to generally healthy kids undergoing elective cardiac procedures. So these are children that are not critically ill. They're undergoing an elective procedure in the cardiac catheterization lab compared to the endothelial cells we get from critically ill children. We collect the guide wire at the time of the procedure and we go for immediate single cell RNA sequencing, again with 10x. And actually we're doing this now with fixed endothelial cells, so it's a little bit easier for us. And what we get are clusters that look like this, and the endothelial cells here are shown in red in the middle. When we zoom in on those, we see single endothelial cells isolated from critically ill children in red, and endothelial cells from generally healthy children in gray. And now, I'll be the first to admit, this is not a lot of cells. And it was a lot of work to get the few number of cells. And there's a lot of problems with this clinical data set. Like, as a researcher, I don't control when the central line goes in, or who gets a central line, or what treatments the patient has had before getting the central line. So there's so much clinical heterogeneity, so much clinical differences. But even with that, in our low numbers, we see significant differences in the transcriptional profiles between the cells from critically ill children and the cells from generally well children. And what we've honed in on are some endothelial cell markers like VCAM, ICAM, E-selectin. These are suggesting these are upregulated in children from the PICU, shown here, with very significant log fold changes and very high p-values, suggesting the endothelial cells are inflamed. And then we see other molecules suggesting that IL-6 signaling may be up. Now, IL-6 is known to go way up in critically ill kids and adults, but this co-receptor right here, OSMR, is also very upregulated, and this stands for oncostatin M receptor. And so we focused on this oncostatin M protein because its receptor and its co-receptor, IL-6ST, are both highly upregulated in the endothelial cells from critically ill patients. And so we wanted to study that molecule, so we got blood from kids from a different study. These are 65 kids with varying degrees of septic shock. So the sickest are shown here in green, and this is oncostatin M levels on the y-axis and hospital day on the x-axis. Children with septic shock have very high levels of oncostatin M for their first 4 days, and then it starts to come down. Children that have mild inflammation are shown here in red, and children that were in the ICU for other reasons besides critical illness or inflammation are a little bit higher, surprisingly, in blue. The difference between blue and red is not significant, but the difference between green and blue and red is significant over the first 4 days. So the molecule is up in the blood of critically ill children, and the signaling is up in the endothelial cells from critically ill children. So we wanted to study what this meant, and this is very complicated because there are many IL-6 family of cytokines. We're interested about oncostatin M, which can signal through the OSMR, which we discovered, and the GP130, which is encoded by IL-6ST. And it can also signal through this thing called the LIF receptor, leukocyte inhibitory factor receptor, and GP130. So GP130 is part of all of these heterodimeric receptors, and all these molecules can signal through different parts of them. Oncostatin M can signal through LIF, and GP130 called type 1 because it was discovered first, and called type 2 through oncostatin M receptors. So you'll see me compare oncostatin M treated endothelial cells and LIF treated endothelial cells, because LIF only goes through the LIF receptor, but oncostatin M can go through both. So later on, we will talk about how LIF is not changing endothelial cells, but oncostatin M is, to confirm that it's type 2 signaling and not type 1 signaling. And we'll come back to that. But you see there are many of these molecules, and so we use that same electrical resistance assay to screen. We did a screen for all of these molecules and found that none of them induce leak. So look at all these lines. They're all clustered between 0.9 and 0, and they're not dissimilar from the They're all the same to the PBS line shown in blue. So none of the other IL-6 molecules induce leak, but oncostatin does. And so we quantified this leak a little bit differently. We looked at the area under the curve, and specifically the area between curves. So we took the area under the oncostatin M curve and subtracted that by the area under the saline curve, and this gives us a magnitude of effect over time, and it's normalized to the saline control. And when we do that, we get graphs like this, where down is more leak because we're taking the oncostatin M, subtracting that, so negative is more leak, and this is less leak. And when we look at all the IL-6 cytokines, only oncostatin M induces leak. So this was very, very exciting to us because this was sort of a underappreciated molecule. I mean, people knew it existed, but people didn't know it was the only IL-6 family member to cause leak. We wanted to look at the type of signaling that was going through this, and so we know that oncostatin M, which comes from essentially every circulating immune cell, every immune cell secretes oncostatin M, and it binds to the oncostatin M GP130 receptor. We wanted to interrogate this downstream signaling, and we did that using Western blots. We immunoblot for signaling cascades, and then we can do gene knockdown in our endothelial cells and do our transendothelial electrical resistance experiments. And so we started off at the receptor. We found that oncostatin M, but not LIF, induces phosphorylation at tyrosine 759. And when we knocked down oncostatin M receptor, we got no leak. And when we knocked down GP130, we got no leak. But when we knocked down LIF receptor, it didn't matter. And so these data suggest that it is type 1 signaling through the oncostatin M receptor. We looked at downstream receptor because oncostatin M and IL-6 are known to signal through JAK-STAT. And in fact, both JAK1 and JAK2 are phosphorylated, tyrosine phosphorylated by oncostatin M. And when we knock down all of the JAKs, we see that JAK1 is required for leak, JAK2 very much significantly contributes to leak, and JAK3 and TYK2, another Janus kinase, are not required. So this suggests that signaling happens through the receptor and JAK1 and JAK2. Now, the JAKs activate the STATs. We know JAK-STAT is pretty common signaling. We tested all of these and found that although the STATs are activated, phosphorylated, by oncostatin M, and less so by LIF, they're not required for LIF, or for leak. So even though there's tyrosine and serine phosphorylation of STAT1, STAT3, and STAT1, when we knock these out, there's no change in the transendothelial electrical resistance, suggesting that these molecules are not required for leak. This is where things get really interesting. So we looked at other sort of less well-described signaling partners for JAK-STAT, and that's the MAP kinase signaling cascade, and oncostatin M activates these signaling cascades. And so we went to probe these molecules, which are complicated and are active in endothelial cells, and through a series of very arduous and difficult siRNA knockdown experiments, we have this, where here on the right, STAT were all not required for leak, and the data is shown here, but SHP2 signaling PI3 kinase through AKT1 and ERK1 are. So of all the signaling molecules, when we knock down ERK1 and AKT1, there is no leak, and the rest of these signaling molecules are not required. So ERK is known to go into the nucleus directly and increase signaling molecules through AP1. AP1, or activating protein 1, is a complex transcription factor with lots of different members. We tested it with a commercially available assay that looks at nuclear localization of these components of AP1, and found that CFOS and CFOS and Jun B are way up, and that CJun is way up in TNF-treated cells. And we found that, in fact, that's true. CFOS, the protein on the top here, is way upregulated with oncostatin M, but not lift treatment. Phosphorylation of CFOS is up with oncostatin M, but not with lift treatment. And the same for phospho-C-Jun phosphorylation. And when we knock out these molecules, we see dramatic effects. So when we knock out CFOS, oncostatin M actually increases barrier function, and when we knock out Jun B, then the leak is reduced. So this is a very surprising finding that we're working on. We're left with this being the signaling pathway that ERK is going into the nucleus to induce CFOS. CFOS is coming out, being acted on by ERK and AKT1, which is inducing genes that may induce leak. We're working on which ones this may be. Probably it's going to involve more next generation sequencing of CFOS knockout cells or ERK1 knockout cells. But again, we want to treat these processes, right? And there are FDA approved medicines that block multiple steps here, right? So there are JAK inhibitors that are well known. They all completely abolish the leak. And these are used in kids for rare conditions, more so in adults, but they reduce this leak. There's things that block the receptors that help, things that block PI3 kinase, things that block AKT1. No FDA approved ERK1 inhibitors yet, but these are all being studied. So hopefully I've shown you that next generation sequencing can identify the changes in endothelial cells that can lead to new findings in how these endothelial cells are signaling, and we can probe those using our cultured endothelial cell models and ultimately block them, setting the stage for testing these therapies or evaluating these therapies in critically ill children and adults. A big problem with that is we have to identify the people at risk for developing capillary leak, which is very difficult, and we're working on that too in a separate study. But I'll pause here and take questions on Project 2.
Nice talk, the Project 2 too. And I wanted to know that capillary leak is not only seen in like shock syndrome, but other some viral hemorrhagic fevers like even dengue and stuff like that. And do you have any guesses that these blocking these? It seems quite interesting that JAK inhibitors might be useful there, but I mean, if it is because of a viral illness, it could be like a double-edged sword. I mean, like, you might be activating viruses because you're dampening type 1 antiviral immunity as well. So,
Yeah, I think in the examples you mentioned, dengue and other hemorrhagic syndromes, there's probably this happening, but there's probably direct injury to the endothelial cells, like the viruses are directly injuring. Because this type of leak, we don't see hemorrhage. And hemorrhage means that red blood cells, which are huge relative to, the proteins in the blood, have to go across the endothelial cell basement membrane layer.
sorry to interrupt. Yeah.
The direct injury to endothelial cells in those syndromes is probably way more impactful than circulating oncostatin M. Does that make sense? Because that's causing endothelial cells to die and slough off.
So, and PI3 kinase is also available, some of them developed for cancers and now used for other situations like APDS. So, have they been tried in these situations in mouse models or animal?
We tested them in cell culture.
Okay.
So, yeah, any drug related to this cascade that you can think of, we've tested. We do a ton of drug screening on this transendothelial electrical resistance assay, and then apolizumab is the best. We're always keen to test new drugs as they come out.
So your model is also a very good model to test the impact of drugs on this leak and stuff like that. Is that correct?
That's correct. Yeah.
Okay. Thank you. Any more questions?
Yeah, it's one of our favorite assays. We love it.
So what you're trying to say is that in situations where there is direct endothelial injury, this mechanism might not be operative. The one which
Yes.
yeah.
Yes. Yeah.
But in dengue there is not. I mean, in dengue there might be just a leak, not I mean hemorrhage necessarily. I mean.
That's true. That's true. And I think the key to that would be to look and see if oncostatin M levels are high in patients with dengue fever.
Okay.
You know,
Thank you.
and you could imagine having a test that says if oncostatin levels are high, then give a JAK inhibitor.
Okay. Sounds interesting.
All right.
So I think you can.
Bonnie, is there time for the third project?
Yeah, you can move on to the third one.
Okay, great. The third project is to take a look at if we can use cell-free DNA in the blood of critically ill children as a biomarker of endothelial cell death in children that are dying with multiple organ dysfunction syndrome. And this is what Boney alluded to at the beginning. Multiple organ dysfunction syndrome is the #1 condition before death in children. Kids don't come in and die of heart failure or lung failure. It's lung failure that causes heart, renal, liver failure, and then they pass away. And so I want to know, is there damage or death to endothelial cells that propagate one organ failing, causing another organ to fail? And it's an attractive hypothesis because endothelial cells are the only cells that are distributed throughout the whole body. And in fact, they're within microns of every organ parenchymal cell. So if there's a process happening in the lungs that's inducing endothelial cell damage or death, that process gets in the blood, that may induce endothelial cell damage or death in the heart and cause the heart to fail. That's sort of the overall hypothesis of this. And this has been really, really challenging because all of the protein biomarkers for endothelial cell damage or death are garbage. And that's because they all are in the same blood. So if you have like venous endothelial cells in your arm that are dying, they'll release contents into the blood that you'll measure, and you don't know if it's from the arm, which we don't really care if vein endothelial cells in the arm are dying. I mean, we do, but it's not going to impact heart or lung function. But if there are capillary cells in the lung or heart that are dying, that may be much more clinically meaningful. But both cells are going to release the same protein biomarkers. And so to date, none of the protein biomarkers have any anatomic specificity of where the death or cell death may be occurring. And so this has been a major roadblock to the field. And we thought we could get around it by using DNA as a precise address, or rather methylation patterns in the DNA to look for this. And so what did we do? Well, we got this notion from these single cell RNA sequencing tissue atlases. So Chan Zuckerberg, the Broad, other institutes are making these huge transcriptional profiles of every human organ, and we found that endothelial cells have unique profiles. And this is probably best shown in the lung, and this is data from the Chan Zuckerberg initiative to single human cell atlas. And what we found was there's restricted expression of genes, and I'm just showing one, this SOS-TDC1, which is sclerosin domain containing protein 1. Who knows what it does, but it's restricted. It's restricted in these cells called aerocytes, which are a specific pulmonary capillary cell. There's about 4 types of pulmonary capillaries, and this is one of them. And these are all the endothelial cells from the Chan Zuckerberg Atlas in the whole body. And you see that of all the endothelial cells in the whole body, only the aerocytes are expressing this SOS-TDC1. So we thought, like RNA is very unstable in the blood, but could we look at the methylation pattern of cell-free DNA and see if there's open regions for aerocyte, for SOS-TDC1, that may mean that there's more aerocyte death. Or to like say it more precisely, like DNA in the blood that is hypomethylated with genes that are restricted to anatomically unique endothelial cells must be due to those specific cells. And so we wanted to test this, and we first had to look and see, is there cell-free DNA in the blood of critically ill children? And the answer is yes. So here is showing yield of cell-free DNA, and this are patient groups. Blue are healthy patients. Red are children that are going for heart surgery before they have heart surgery. So heart surgery is a major insult to the heart and the lungs, and children going in beforehand don't have injury, but children coming off cardiopulmonary bypass, which is also known as the heart-lung machine, children coming off the heart-lung machine have profound injury to their heart and their lungs. And so we can get blood from children before the heart-lung machine and after, and we're thinking, man, this would be great to study. And so before, there's none, and after, there's a lot of cell-free DNA. And then this graph is looking at children, the pairwise comparisons, and we see it goes up for almost everybody except for this patient in black here. I'm not sure what's going on with them, but in general, there's more cell-free DNA in the blood of sick children after bypass. And we wanted to look for profiles of specific genes, and this was done using a twist assay, to look at specific genes. Could we sequence open profiles for specific genes? And in fact, we can. This is looking at specific genes here, and this is looking at percent change in methylation. And we find between 5 and 15% hypomethylation, right? These are all negative change in hypomethylation in genes for general EC, for lymphatic ECs, for lung capillaries, right? This is the SOS-TDC1, lung aerocytes, is a specific capillary. So this is just suggesting that there's more DNA open in those areas after bypass, suggesting that there's injury to those specific cells. This is the first time that there's such an anatomically precise biomarker of endothelial cell injury and death. And so we want to elaborate on this, we want to do more than this, and so we got single nuclear, so we want to look at the whole methylation profile, but that's super complex, and we need a decoder ring. We need a way to decode the methylation profiles we're seeing in the blood versus before and after injury, because we picked out this SOS-TDC1 gene, but there may be better genes. There may be better genes that we discover from looking at the whole methylation profile. But to do that, we need a single nuclear methylation sequencing, and we do single nuclear RNA sequencing and methyl-seq on healthy pediatric heart tissue. And the goal of this is to identify specific endothelial cell populations using the RNA-seq, and then identify or decode the same methylation, the methylation patterns that match that RNA sequencing in nuclear and methyl sequence of patients, and ultimately to the blood of critically ill children. And so about this strategy can go to Francesc on using the of endothelial cells, code the methyl sequence, and then look for that in the blood. So we want endothelial sequence here, what sequencing matches the cell cluster. And so this process is complex. We can look at islands or hypomethylated regions, specific for endothelial cells or CDH5, PTP are restricted phosphatase, endoglin is an endothelial marker. And when we look at this cluster here is endothelial cells. The cluster here is definitely cells. And this cluster here, endothelial cells, but probably not. We're still working on it. Using the single nuclear RNA sequencing by single cells in the single nuclear methyl, and then we can take these profiles and interrogate the blood of critically ill children. But we wanted to make sure we had enough cell-free DNA. These are kids after with septic shock, so the methyl levels are even, right? And if we look at kids in septic shock, we, as they have multiple organ dysfunction syndrome, and when they have no multiple organ dysfunction syndrome, low. So we look at patient 1 in purple and patient 2, and we see, Franceska's done this analysis, higher proportion of endothelial in the cell-free DNA of purple and red. So this is looking at the fractional cell DNA. In the total, we see very little in healthy control, or and a lot when they have from here. So this project is on getting better resolution, both with our sequence, but hopefully I've impressed upon is up in kids that are sick with bypass or more, and that endothelial cell patterns can be detected. And we're going to write a grant to try and see if the proportion endothelial specific organ failure, or to say it differently, there was pulmonary capillary endothelial cell death, would that predict the duration, onset, or severity of lung injury? We think it would, but we're going to write a grant to find out. So that's all I have. I'm happy to take questions on Project 3 or any of the projects.
Any question for anybody? Oh. Anybody wants to ask any question? Okay, great. Thank you so much, Rick. I think it was a great presentation, and thank you so much. You know, Rick?
Yeah.
Yeah, just yeah. So thank you so much, Rick. It was indeed a great presentation, and I hope people learn a lot. And I'll see you in New Haven soon.
Yeah. Okay, great. Well, bye everybody.
Bye-bye.
Thanks for staying up late for me.
Say hi to Francis from me.